Hello? Can you hear me, the online audience, please? Okay. Hello? Okay. Yeah, thank you so much. Sorry for the inconvenience. I want to go through the schedule for today. So we're going to start with the announcements and go through the first talk of the day with the introductory trainee talk, and then move on to the future talk at 6:20, and the event is going to end at 7:20, and the social is going to end at 8. So I want to go over the organizing committee as well. VEMBAG is led by 3 faculty members: Dr. William Shao, Dr. Emily, Dr. Faraz Haj, and wonderful volunteers Carolyn, Jonathan, Varia, Madeline, Yeren, Emily, Matty, and myself. And we have 3 more VEMBAG events for the year: January 15th, February 19th, and March 19th. So please be updated on those with our mailing list as well. If you're interested in being up to date with VEMBAG, you can go to our website, go to the contact tab, and under the mailing list section, you can fill out the form that is circled here to get the mail updates. Also, you can follow our calendar and social media accounts to keep yourself updated with VEMBAG events. Here are the QR codes for those. And I'm sorry. If you want to suggest a speaker for the next season, you can also go to the contact tab, and under the suggest a speaker circled here, you can suggest new speakers for the next season. And we are sponsored by Graduate Studies in Bioinformatics at University of British Columbia and Simon Fraser University, Genome British Columbia, SFU Big Data Hub, Michael Smith Laboratories UBC, Langara College, and Stem Cell Technologies, Vancouver Prostate Center as well. And VEMBAG is affiliated with ISCB, our sister bugs, Monbag and Torbug. And if you want to ask questions in the chat, please do so, and a volunteer will voice it for you for the online audience. And we are going to first start with Irvin Ng's talk. Irvin is a master's student in bioinformatics from Cortaro Lab, UBC. And his talk is going to be about predicting sustained remission and maximal disease severity in pediatric Crohn's disease using machine learning.
Everyone. Wait. It's not a. Oh, screen?
Yeah.
Cool. All right. Hi, everyone. As Berkeley mentioned, my name is Irvin, and I'm from University of British Columbia and PC Children's Hospital. I'm a master's student co-supervised by Drs. Bruce Valens and Dr. Keegan Korthauer. And today my talk is going to be on my project, which is predicting sustained remission and maximal disease severity in pediatric Crohn's disease using machine learning. And so quick background information about my project. So IBD, or inflammatory bowel disease, is a group of chronic inflammation conditions that affect the GI tract or the gastrointestinal tract. And this can be broken down into 2 main subtypes. The first subtype is Crohn's disease, which is characterized by these kind of patches of inflammation throughout the GI tract. And then the second type of IBD is ulcerative colitis, which is characterized by these continuous patches of inflammation and is limited to only the colon. Now, IBD is idiopathic, but current literature suggests that it's a multifactorial disease where it's influenced by an interplay of different factors such as environmental factors, your diet, microbiome, etc. Additionally, the clinical manifestation is actually quite heterogeneous, where patients can present with a whole range of symptoms, and as well the disease progression itself is also very heterogeneous. And approximately 15% of all cases are pediatric onset, where patients are diagnosed as children, and approximately 2/3 of these pediatric cases are Crohn's disease, making this the more prevalent case. And so pediatric patients is a little bit special because they present with a more extreme and extensive and severe phenotype compared to adults. And with all this inflammation, increases their risk of malabsorption and malnutrition. And because they're children, this puts them at higher risk of developmental stunting, growth delays, etc. I also mentioned that disease progression is very heterogeneous. So for, sorry, the pointer's not working. Give me 1 second. So in some cases, the patients come in and they go into, they have active inflammation, they go into clinic, they get diagnosed, they get treated, and they undergo remission. But the problem is that a lot of patients actually don't achieve sustained remission, where they undergo treatment, they get remission, and then they have these kind of cycles of flare-up in their inflammation. And the problem is with each consequent flare-up, puts them at a higher risk of complications, ultimately needing surgery. And so there's this window of opportunity here where the current perspective is changing, where it suggests that for these patients at risk of having more severe disease or having at risk of having these recurring flare-ups, if we treat them with more aggressive therapy earlier on, we can improve their outcome. But the problem is we don't really know which patients are going to be at risk. And so my project is using baseline clinical and microbiome data at baseline, trying to predict basically 1-year disease outcome in these pediatric cases. More specifically, I'm predicting 2 first-year outcomes. So the first outcome is sustained remission versus non-sustained remission, where I predict whether patients stay in remission or they have these flare-ups in 1 year. The second prediction task is predicting disease severity. At their worst disease state, are they having severe disease or are they having a relatively milder disease? And so the data set that I'm working with is a data set from KidsCan, or the Canadian Children IBD Network. And this is a multi-center prospective cohort of children across Canada. And the data set itself, has around 1,500 patients. And as expected, the majority of these patients have Crohn's disease or IBD unknown favoring Crohn's disease. And I wanted to use patients that had complete baseline clinical, biochemical, and linked microbiome data. And so ultimately, I had my final sample size was 86 patients that had all this complete data. Using these patients, I made 3 data sets: clinical data set, a microbiome data set, as well as an integrated data set, which has both microbiome and clinical data. And then for the study, we built models using the 3 data sets. And essentially what I did was I used a repeated nested k-fold cross-validation framework. What this does is that we have 3 outer validation folds, and then we also have 3 inner folds, where the inner fold is used to do your training and validation, and the outer fold is used to do testing. And this is done repeatedly to give us a robust estimate of model performance. And then I built models using 9 common algorithms, ranging from logistic regression up to decision tree. And finally, we have 2 main outputs. The first output is looking at the top informative features using an algorithm called SHAP, or Shapley's explanations, additive explanations. And the second output is a comparative ROC plot, just looking at the AUC of the models and as a metric of their performance. So looking at our first prediction task, once again, this is looking at patients predicting patients that will stay in remission versus patients that have a disease flare in this 1 year timeline. Here I looked at whether the gut microbiome at baseline or the clinical data at baseline had any significant associations in the univariate analysis. And what we see here is that there's really not much signal or predictive signal when you do a univariate analysis, where, for example, looking at the differential abundance analysis of the microbiome, there's nothing standing out in terms of significance, and likewise for the clinical data. But when we move beyond univariate comparisons and use machine learning to look at the data sets as a whole to predict sustained remission, here we see a different picture. And so when we compare the mean AUCs of all the models, we see that the models trained on the integrated data set here either outperform or perform similarly to all the models trained on IDA data set or data modality alone. In particular, the logistic regression model achieved the highest AUC of 0.76, or sorry, 0.763. And this demonstrates overall that integrating the clinical and microbiome data yields the best predictive performance for predicting sustained remission in pediatric Crohn's disease. And also highlights the value of combining data sets to capture potentially complementary signal. And taking this one step further, to better understand what features are driving our predictions, here I show the top features as measured by SHAP. And we see things, for example, like initial therapy, Adoribacter, or Haemophilus driving predictions of non-sustained remission, whereas things like proximal disease location, Eubacterium elegans, or Lactobacillus are driving predictions towards sustained remission. And this is actually validated in literature, where, for example, things like Haemophilus has been shown to be a microbe that exacerbates Crohn's disease because it disrupts the epithelial barrier and integrity of your GI tract. On the other hand, things, for example, Lactobacillus, this has been shown in literature to be protective against inflammation because it produces something called a short-chain fatty acid. And then now looking at the second prediction task, once again, this is predicting patients that at the worst has severe disease or has mild disease. Once again, when we do univariate analysis, this reveals very minimal predictive signals. Once again, looking at the microbiome or the clinical data, we don't see anything that's statistically significant between the classes. But when we look at the machine learning models, once again now we see a different picture. So previously, in the previous slide, the microbiome didn't have any strong signal for differentiating the two classes. But in this case here, the microbiome or the model, the Gaussian naive-based model that's trained on the microbiome actually outperforms all the other models, and an AUC of 0.0801. Interestingly as well is that there's a very poor clinical performance. And so this suggests that overall, the microbiome alone may be a sensitive early indicator of the inflammatory process. And combining the two actually drops the performance, most likely because the clinical data is actually introducing noise that overwhelms the microbiome signal. So looking at the top model, here we see that two strong features are driving predictions. So Clostridium is driving predictions towards more severe disease. And this is validated because a very well-known species of Clostridium, Clostridium difficile, or C. diff, has been known to be pro-inflammatory in Crohn's disease patients, whereas Coprococcus, on the other hand, is known to be protective against inflammation. And so the key takeaways here is that machine learning can help clinicians tackle two major questions in pediatric Crohn's disease. One, first, is who will sustain remission versus relapse? And second is who will have severe disease? And then here we show that integrating the microbiome and clinical data together provide better predictive value for predicting sustained remission. And overall, logistic regression and Gaussian naive-based models generally perform the best in our data set. And that concludes my presentation. I'd like to thank my supervisors, my project advisors, and my committee, as well as the members of my labs and the KidsCan team and study participants. I'm happy to take any questions or comments. Thank you.
if you have any questions, please use the microphones in front of you and click on the red button. So it would take turns based on your order of pressing. So if you have any questions, please feel free to ask right now.
Hello. Hi, Irvin. I don't know anything about the microbiome stuff. I work on other things, but could you talk a little bit about like how granular the species are that you're looking at in those plots? You mentioned that one of them was extremely predictive, another one was extremely predictive the other way. How granular is that?
Sorry, can you say that a little bit louder? I can't actually hear what you.
How granular are the species that you're looking at? So one of the microbiome or one of the small bacteria that you're looking at was extremely predictive of bad disease. Another was extremely predictive of the other way. How granular are these? Is this at the species level? Is this below that? Yeah, could you talk about that?
Yeah. So this is, first of all, this is genus level. In terms of how regular, I assume you're talking about like how, for example, patients that have more severe disease, how like often do they actually have, for example, Clostridium in their microbiome? Is that what your question is?
No, I was more wondering, like, is the genus level, is that what matters?
Oh, I see. Yeah, so in the ideal world, it would be species level or strain level. In this case here, I'm limited by the fact that the network used 16S. So with 16S sequencing, the highest or the highest level of resolution that I'm confident with is genus level. So that's why I'm working with the genus level microbes here. Sorry, in terms of regular, or did I answer your question or? Oh, okay, perfect.
Sorry, kind of a follow-up question to that. I saw in the previous analysis, you said Bifidobacter is a predictive of less remission, but isn't that also a short chain fatty acid producer?
Sorry, which?
For the other one, the other graph like that, Bifidobacter, that's a genus that is often associated with positive disease outcomes, right? They're short chain fatty acid producers.
Yeah. So that's the problem with genus level. It's hard to tease out because even with the genus level, there are certain species that are beneficial and there are certain species that are detrimental. Like for example, like you said, there are certain species of Bifidobacterium that are short chain fatty acid producers. It's hard to tease out exactly which species is driving this prediction. Just that it seems to be driving predictions overall. Bifidobacterium seems to be driving it towards non-sustained remission.
Thank you very much for the talk. So back in April this year, a paper came out in Nature that showed Colibactin, I think was the name of the bacteria, had a link to colorectal cancer. So it was basically a mutagen and it created some sort of an inflammatory environment that increased the risk of colorectal cancer. I'm just wondering what your thoughts on that, or if you think there could be some sort of a link between the species that you've identified and worked with the future risk of colorectal disease, including cancer.
Yeah, I think that's a very good point. So one of the things with pediatric Crohn's disease, or I guess pediatric IBD in general, is that it puts you at a higher risk of colorectal cancer, just because this is a lifelong disease. When you have all this chronic inflammation, especially for example in the case of that Nature paper, which shows that microbe driving inflammation. When you have all this long-term inflammation, it puts you at higher risk of colorectal cancer. So I'm not surprised to see that. And another thing I wanted to point out as well is that there has been a lot of studies shown where certain microbes drive your risk of colorectal cancer. So I would agree with that paper.
Sorry, just one follow up then.
Yeah.
Did Colibactin come up in your analysis? Do you ever remember coming across it?
Not specifically. The thing is with microbiome, there's a lot of heterogeneity. Not even between different populations, but even between like me and you, for example, our microbiome is very complex. And so what might be significant in one study is very likely not going to be also significant in another study. As well, it should be noted that this study has 86 patients, so that also, yeah, it's hard to tease out a lot of significant microbes there. Yeah.
So I'm just looking at the top feature up there. Can you tell me what it means? What does the aggressive therapy look like, and what would a non-aggressive therapy look like?
Yeah. So that's a great question. In this case, I grouped the initial therapy as aggressive or non-aggressive. So in this case, aggressive would mean things like a steroid or something like an immunomodulator or a biologic. But usually with patients that have more severe phenotype or very high degree of inflammation, the first line would be corticosteroid. The problem is that a lot of times corticosteroid alone is not enough, and we have to wait until they fail that, and then we escalate it up and up and up. And that's also one of the point of this project is to see, hey, this person is likely going to have a very severe disease. So can we start them with things like, for example, a corticosteroid and something else? And then something that's not aggressive would be things like an antibiotic or something called EEN or exclusive enteral nutrition, basically a liquid diet. That has been shown to be very helpful in inducing remission. But yeah.
From Angela in the Zoom. Great talk. Can you please elaborate on the cross-validation design in terms of how you group data into folds and whether that reduced data leakage?
Yeah, so in this case here, I split the data into, as you can see here, I split the data into an outer loop. And then, so within the outer loop, so there's a training fold and a testing fold. And within the training fold, I then further do the split this into 3 folds where we do the training and the validation. This is done 3 times, i.e. it's repeated just to get a better estimate of the best parameters. And then the actual evaluation of the whole model is also done 3 times to get a robust estimate of the model performance. Just making sure that the metrics or the performance is not due to some lucky split. Yeah.
Hi.
Yeah.
Thank you for your talk. So I have a question. Maybe I missed it, but how many features you have for your models?
Yeah.
I remember you.
I didn't really go into it because I only had 10 minutes. So in this case here, the clinical data set had 34 features. The microbiome data set, it only had genus level taxa as well as the diversity metrics. So in this case here, the microbiome data set had around 110 features. And then so integrated, it was around 140, 150. Yeah.
Mm-hmm. Yeah, and these are like continuous values in your.
Not particularly. So, for example, for the clinical data set, you have things that are like gender. So that's binary. But they also have things like lab tests. So, for example, certain inflammatory markers such as C-reactive protein, and that could be continuous. Yeah.
Mm-hmm.
All right. Thank you, everyone.
Thank you so much, Irvin, for his amazing talk. And give me a second, please. Now we're gonna see Dr. Chadi Saad-Roy's presentation. Dr. Chadi Saad-Roy started in September 2025 as an assistant professor at the University of British Columbia, split evenly between the Department of Mathematics and the Department of Microbiology and Immunology. He's a core member of the Prepare for Pandemics through Advanced Research in Evolution, Prepare in short. research cluster at UBC, and he's a member of the Institute of Applied Mathematics and the Biodiversity Research Centre. Saad-Roy's interdisciplinary background melds biology, mathematics, computation, and statistics. And thank you so much again for coming.
Oh yeah, nice. Awesome. And now we turn this on. No, I don't think so. Oh yeah, it's closer. Can you hear me? Yeah, okay, good. Is that good, or you can hear? Yeah, oh, interesting. I can't hear the echo, so it's very strange. Oh, you speak more into the mic here. Let's put. Is this better? Is this better? Can you hear now? How's this? No? Kind of, not really. No, still not. Okay, there we go. Now we can hear it, right? No. It's not. Sorry. Let's try it here. Okay, how's this? Yeah, now you can hear. Okay, good. So we're trying, yeah. Took a little while. Sorry about that. Yeah, thanks for having me. It's awesome to be here and to see your community. I didn't know this existed until Bjarne emailed me, so I thought it was really cool that you guys come together every month. Hopefully it'll be I think it'll be a little bit different than your typical bioinformatics talk, but hopefully I do have I forgot to put in the in the biography, but I have a PhD in quantitative and computational biology, so hopefully it fits within what you guys are interested in as well. So I thought I'd start off very broad with some motivating questions that motivate me, have motivated my work over the last however many years. In particular, I'm thinking about how do susceptibility and immunity influence infectious disease dynamics. So this is thinking about infection, thinking about hosts, and how they together interact. A little bit diving deeper, what's the interplay between evolutionary, immunological, and population level processes for infectious disease dynamics? How do these things across scales affect what we see? And finally, how do social human behavioral dynamics shape epidemiological trajectories and vice versa? So before I start introducing, you'll see there's going to be flavors of each of these throughout the talk, but I thought I'd introduce to you what I mean when I think about disease dynamics and what I mean by cross scales. So there's going to be a slightly busy plot here, but we'll walk you through each set of the figure. And it's a schematic. And by the way, the colors stay mostly the same throughout the talk. So once you see one color type, then you know that this is consistent throughout the talk. So you don't have to continually learn new ones. So the very base here, we have this emergence of new strains, right? So where do new strains come from? And I think it's helpful to cast this in terms, if you think about SARS-CoV-2, a lot of these examples, you can think about that. And there's examples at each level for that, right? So emergence of new strains. So all of a sudden we had Delta, Omicron, et cetera, right? So this became quite relevant pretty quickly, right? It had an impact on public health. This is at the molecular level. Now, what about slightly up higher? We have cellular and tissue transmission. So here we're thinking about how tissue tropism, how does a virus or pathogen transmit through tissues and cells? And here I put the little lung because that might be tissue tropism. Maybe some viruses are better at infecting certain organs or certain parts of the body than others. And again, here you can think about Omicron. Part of the reason it was successful is thought to be because it's slightly different tropism, right? So there again, you can see there's a COVID example. Then you move on again, still within a host. Think about within host kinetics. So here you have these, right? So you have this within the host. Oh yeah, okay, you can't see the laser anyways. But yeah, so you have within the host the viral loads. So this is the infection takes up, and then eventually the adaptive immunity, the thing beside it. Now let's see if I can show you on this here, yeah, right here. So this eventually kicks in and then controls the pathogen, right? And then your viral loads go down, and then you recover, right? So then adaptive immunity is there. And then if you think about that, that's at the individual level, but that also impacts when you get infected, right? So if you're an individual that's first infection, you get infected, and then you transmit to people along this spectrum from that are fully susceptible, that never seen anything, or that are fully immune. Well, if you're contacting someone that's fully immune, then they're not going to have a transmissible infection, right? But then somewhere in between, you get a secondary or breakthrough infection, and then the characteristics might be a little bit different than what you saw initially, right? And then if you have a fully susceptible person, then it's likely to be quite similar to what you had before. And here in this corner, there's this immunocompromised box with prolonged carriage. So this is the idea that some individuals that are immunocompromised might hold these infections for a long time and generate new sources of variants, right? And so this kind of takes us back to the first bit, right? So these are all interconnected levels. And then you can think about immunity, right? The strength and duration of immunity. So an individual gets infected, recovers, and then they're immune for some period of time. They have this strain transcending immunity. And then you can either have two processes, right? So a pathogen-driven process, a drift, antigenic drift. So those surface proteins change. You can get reinfected by a slightly different pathogen than what you saw before, right? So the pathogen's drifting. Or your own immunity can wane, and you can get reinfected by what you saw before, right? So then the immunity is eroded in either case. So you have the strength and duration. Now, if you add up enough people that are immune, you can get this community immunity, right? So now this is kind of like, I think was very popular in the news. We have to reduce R naught, right? R naught, this basic reproduction number concept. So if you have enough immunity, you prevent invasion, right? And if otherwise, if you don't have enough immunity, then you can get new invasion of pathogen, right? And infection spread. So this is again a community. And now if you think about the globe, then there's global spread, stochastic persistence, and stochastic extinction, right? And so now all of a sudden you can have regions with strong immigration. Maybe it's easier for infections to go across, and maybe with weak immigration it's harder. And so this region stays with no pathogen for some time. And then a new strain comes about, and then it spreads, and maybe it spreads and persists, or maybe it goes extinct, right? And that's a new strain. So we're back to asking about where the new strain. So we're closed the loop. And in the middle, you'll notice this actually wasn't part of the original schematic, but I modified it because I think this is important now. Individual behavior, right? So do individuals choose to interact? Do individuals choose to adhere to some non-pharmaceutical intervention, mask wearing, social distancing, et cetera? Or do you choose to get vaccinated? And I think that can have an impact at all the levels. Right? So I think this is a complex system. You can think about that together. So one of the first questions that you can ask, and a lot of this will be, some of the following bits of the talk will be cast in terms of COVID, but you can think about this in terms of any emergent pathogen. Right? And one of the first questions you ask when you have an emergent pathogen is, what are its dynamics as it transitions to endemicity, right? When it's endemic and it's in circulation. And so for SARS-CoV-2, we were looking at this in terms of how does this depend upon individual immune response, right? So initially, everyone is susceptible. You get infections, but eventually people recover, right? And then now all of a sudden you have all these recovered individuals, and what happens? Well, these recovered individuals, they can potentially stay immune for a long time, or maybe they can wane to be back to being completely susceptible, or maybe somewhere in between, right? This is this depiction here. And so the strength and duration of immunity may be relevant, right? So we can examine how that plays out. What about vaccine hesitancy? What about non-pharmaceutical intervention? So this is if you reduce transmission, right? So you implement some non-pharmaceutical intervention, you have lower transmission. How does that affect the medium-term dynamics? And what about seasonality? So we know a lot of infections are seasonal drivers. You have some more infections at certain times of the year than others. How does that play in to these complexities? So we investigate this, and this is the paper and the reference. And I'll just give you a very brief highlight of this work. So the basic modeling framework. So this is for those of you, I think because of what we lived through, you're probably aware of the susceptible, infected, recovered kind of models, these SIR models. Yeah, so I see people nodding. So basically you have that on one end, right? So susceptible, infected, you're recovered, and you have lifelong immunity. On the other end, you have this SIRS right here. So you're susceptible, infected, recovered, and then you wane back to being completely susceptible. And then, but in reality, a lot of pathogens probably somewhere in between, right? Your first infection is quite a bit different than your subsequent ones. So we can leverage this SIR bracket S model from Morris et al. In a paper in 2015 to look at that. And so what this model says is you're born into this primary susceptible class, you get infected into the primary infected class, you recover to the R class, You wane into the secondary susceptible class. Then you could get infected, IS, secondary infection. And then again, for simplicity, you assume that these go back to the R class. So secondary, tertiary, quaternary, et cetera, are all the same. Okay. And now, so there's a couple parameters here that are important. So the beta is the transmission rate. The delta is the rate of waning immunity. The gamma is the recovery rate. And now alpha, which is here, is the relative transmission rate of secondary infection. So if you have a secondary infection, how transmissible are you relative to the primary one? And epsilon multiplies the beta down here, you see. That's the relative susceptibility to reinfection. So you're in SS, what's your likely, how susceptible are you compared to someone who's never been infected? Now, if you set these to be 0 and you set delta to be 0, then you can see you can never leave the R class, so you get an SIR model. And if you set them to be 1, then you're as susceptible and as transmissible in secondary susceptibility and secondary infectivity, or secondary infection. And so you can just add these up as primary ones. So you add these up and you get back to your SIRS model, right? So secondary infections, tertiary, quaternary are the same as, okay. So this actually, now you can use epsilon and alpha to interpolate between the two, right? So you can go from one end to the other. So you can look at that. And the other thing is I told you seasonality might be important. So you can put that right into the beta, right? So beta transmission can be seasonal. And that's also where you can reduce the non-pharmaceutical event. So you say, okay, now for this period of time, transmission's down. Okay. So these are the model equations in case anyone likes to see some dynamical system. Well, there it is. And you'll see the beta here is time varying. So that's why the beta of P there for the seasonal and non-pharmaceutical interventions. So how does this all play out? So there's a few parameters here, and we can actually use related coronaviruses to benchmark seasonality. So we can use that. So we get a seasonal transmission rate. And then you can ask questions about how important is epsilon. So epsilon is relative transmissibility to reinfection. So this is a little busy, but I'll walk you through the panels. Basically, there's 3 scenarios here. On the left, you have this long period or slightly longer period. So the bottom, no, my cursor is lost again. But the bottom icon illustrates dark areas where the non-pharmaceutical intervention is implemented. So you reduce transmission, you fix it, reduce it by some amount. And then the gray is it's relaxed. We have some period, non-pharmaceutical intervention. The second scenario, the middle one, you have a shorter period. And then the right one, you have a short period, you relax, and then you have a shorter period again. Okay. And then the top panel, so now you have 3 axes. And now if I can find my cursor, it'll be pretty great. Okay, so here you have the time on this axis, the epsilon value on that axis. So remember, epsilon towards 1, you're as susceptible to reinfection as a primary infection. And epsilon towards 0, you're like SIR-like, so your immunity is very good, right? And then on this axis, you have the fraction of the population. The colors are just illustrating different values of epsilon, and the line type is the primary or secondary infection. And so what you'll see is the first peak, right, looks the same across the values of epsilon, right, for this scenario. That makes sense. No one's been infected. People are fully susceptible, so immunity shouldn't have an impact, right? Now it gets kind of interesting as you look at progression and you look at the second peak. And now all of a sudden you have this non-monotonic behavior, right? Whereas your weaker immunity increases the size of the second peak and then decreases it. It's a little bit strange, right? So it's not obvious what's going on here. If you look at the other scenario that I just described before with the shorter period of non-pharmaceutical intervention, you have the opposite relationship. Intermediate immunity minimizes the peak. So what's going on here? So essentially, you have this seasonal cycle and this interaction. So you're basically, because you have this seasonal changes in transmission, you get a delayed but bigger second peak, right? Susceptibles can accumulate, and then you get a bigger second peak. The point here being, figuring out what epsilon is kind of matters, right? Because if you don't know what it is, you can vary it with small degrees and get fairly big differences in outcomes. So I think this illustrates that we need to measure what epsilon is. And this was at a time when we weren't really thinking about this characteristics of immunity, right? Or using SIR models to try to infer this initial growth rate. But this convincingly shows that you need to know a bit more than that for the medium term. So we were really interested to generate these medium term landscapes. So what might happen in the medium term? And now, for someone who's been paying attention, you might fault me for not including vaccination so far, right? So the previous model don't have any vaccines. But at the time, we didn't really know what vaccination looked like. But we actually incorporated vaccination just to see what might happen in a very straightforward, simple way. So now we have this V class here. The V class individuals are immune, and we were assuming that the population's getting randomly vaccinated, and those that are susceptible get bumped into this class. And then their immunity wanes at potentially different rate, delta vax, right? So the average time you're in B class, 1 over delta vax, which is different than natural or infection-induced immunity potentially. And now we can, again, do a little bit of analysis, find the minimum vaccination rate for eradication, and then you can look at synoptic landscapes. So here we've got area plots on this bottom panel, and then the top is some plot on the severe cases. Again, we're projecting forward over time, 5 years. The left column is situation with poor immunity. The right column is situation with robust immunity. The top row is no vaccines, and the bottom row is vaccines. And we assume here when we have poor immunity, it's poor transmission immunity. So the duration is short, the relative susceptibility is high after immunity wanes, and the clinical immunity, which protects you against these severe cases. So you have to assume something about primary and secondary cases. Here we assume a pessimistic scenario for secondary cases. And in the robust immunity, we assume that the clinical immunity and the transmission immunity is robust. So in the poor immunity case, what do you see? Well, you got a lot of recurrent severe cases, right, over the medium term. And basically it's a pretty negative outcome, right? You never really get out of it. In the very positive case, where you have this robust immunity and a good vaccine, then you get near case elimination, right? Somewhere in the middle, you have this situation where you've got infections, severity goes down because now there's secondary infections, and they're still prominent because immunity is rather poor. So this kind of shows you that there's a large range of outcomes just by moving about these parameters, these immunity parameters, and what if you have a vaccine or not. So this is a very simple way to consider vaccination. And now again, you'll say, but actually there were multiple doses. So initially there were 2-dose vaccines, and then there were questions about whether to extend the space or not between them, right? And then there's different arguments about epidemiological arguments, evolutionary arguments. And so we realized we have the models to take a look at what happens in these situations. So now we extend the base model to consider now, instead of now maybe we can decide whether or not this is simple anymore. It is the title of the talk, but I think in my opinion it's still simple. Some may disagree. So you have the base model, right? So you'll see the outline here: SP, IP, R, SS, IS. Well, then the V class splits into 2, right? So now you have dose 1 in like green, and then dose 2 in dark green. And then each have their own secondary susceptible class, SS1 and SS2. And then each with their own infection class, IS1 and IS2. So each with their own epsilons, right? And stuff all carries through. And then you have IVs. So this is where infections during vaccination. So the vaccine didn't work. So now you have to also consider this omega here, which is the rate that, so 1 over omega is the average time between V1 and V2, right? So this is the average dose distance. And you can link that up to the vaccination rate, which is this parameter nu, same as before. So this is what's illustrated here. So if you have the optimal dosing space, L-opt, then you're vaccinating at exactly half the rate than if you only were doing 1 dose, right? So the idea being here, I can vaccinate Will and Bardia, or I can do that, or if I know that Will's going to come back in 4 weeks, then I have to hold the other dose back, right? And give it to him in 4 weeks. So I can get double the rate if I'm doing the 1 dose. Okay. And so you link that up through this kind of this relationship, and then this is what and then the evolution I'll discuss in a second. And then this is the reference for people interested. And so you can first examine the epidemiological outcome, right? So what happens epidemiologically? And we can now I told you the synoptic landscapes, there's 4, now each become many more. So I took out the one that I think maybe is the most relevant one for us now in retrospect, but you can look at the paper and look at all of them. But here's the one where we have less robust natural and vaccinal immunity and shorter and weaker immunity following 1 dose. And here you can see in a 1-dose strat, so the rows are the dosing strategy. So 1 dose, 24-week interdose, 12-week interdose, and then the recommended 2-dose. The top is just a cartoon for the rollout. how many individuals are vaccinated. The middle panel is this cases in red, and then again the severe cases in solid in dash black. And this is again assuming some assumption about how vaccination protects you. And then the bottom panel is the area plot with all the different colors. And now, just in case I went through too quickly, they're all here now, right? So you can see all the colors, and they all show up as different parts of the area. The whole population is one, and you split the population over time. So what can you see here? So if you look at the one-dose strategy, because in the simulation we're vaccinating, we start vaccination right when this is starting to take off, right? You really do reduce this initial peak, right? If you compare the one dose to the two doses, you see that you get really good bang for your buck at that level, right? You're reducing, snuffing this first peak. The problem is in the one dose strategy, you get a lot of these orange individuals, right? And now orange is the waned one dose immunity, and now waned immunity here, people are relatively susceptible. So then that triggers pretty high second peak, third peak, and fourth peak, right? You have medium term infections that are elevated. In the case of the two dose recommended dosing, you don't have that, right? You never get this buildup of orange. And now actually, if you look at the intermediate cases, you can reduce the initial peak, but also don't have as big later peaks, right? So as the 24-week or the 12-week, and you can see the orange, there's a lot less accumulation of those, right? So what's going on? Well, those individuals are now able to, you're basically reducing this elevated susceptibility by ensuring that they get their second dose later on. So somehow, in our minds, this is probably the answer, right? So somehow you just ensure, initially you roll out the one dose, and then when you have high enough, you're not in emergency settings anymore, you ensure that they get their second dose, right? Even if it's a lot later. So this is kind of interesting. And then we can think about the evolutionary outcomes, right? So the evolutionary outcomes, this is a lot more hypothetical. We don't really know what's going on, but we can make like a zeroth order model, the first kind of attempt at this kind of what might happen. So this is going to be work built on this file dynamic curve. So I'll introduce what the file dynamic curve is. It was introduced in a landmark paper in 2004 by Brian Grenfell and co-authors. And basically, the idea is you have two key features here, the strength of selection and the viral abundance, right? So as your immune pressure goes up, the strength of selection goes up, right? You have more and more selection. And now as immune pressure goes up, the viral abundance goes down, right? There's less and less virus. So if you have no virus, there's no potential for evolution. If you have no selection, there's no potential for evolution. Somewhere in the middle maximizes it, right? It's about like here. So that's why so this is this file dynamic curve. And it's been very influential in this field. It kind of launched biodynamics as a field. And you can think of flu, often we used to think of flu as sitting on top of this plateau, kind of around here. And anyways, the original 2004 paper, if you're interested, will really classify different pathogens according to these patterns. So what we're interested here is to look at three different scenarios. One scenario, this left one here, we have low potential variation, right? So all of them are in this, so the black line's the file dynamic curve. And now we're all in this area where we have very low variation, very low potential adaptation. The scenario two, you have low potential with natural immunity or two-dose immunity down here, but the one dose is high. At scenario three, they're all very high, right? And then you can just project forward and look what might happen. So I just, again, take one sample to show you that you can do this. You can look at the paper to see more scenarios. Again, the dosing regime on the rows, the columns, and the scenarios are in the colors. And you can see if you're in this scenario, it doesn't really matter what dosing regime you're in, right? If you're in the black scenario, low potential viral adaptation, all three look very similar. Scenario two and scenario three, all of a sudden the dosing regime really does make a difference, right? So somehow it's good to have an idea about what might happen in different cases. So then we took this work forward in a variety of directions. I don't have time to go through all of them, but I'll just highlight a few key findings. So one is that vaccine nationalism can have negative epidemiological and evolutionary outcomes. So this is this idea of sharing or not sharing vaccines with different regions. and accumulating immunity. So far I showed you have a primary infection, a secondary infection, and secondary is the same as tertiary, quaternary, et cetera. But what if they're potentially a bit different? So exactly, so you can model that, and this is what this work has done in this paper. And then we also look at long COVID, right? Different infections may have different risks of long COVID, so you can look at that if you're interested. And then if you have severity blocking immunity, so what if you have different time scales? So your immunity can wane, but then you still might be protected against severe disease. Then what if you can wane again into a separate class? So you have 2 time scales there, and that can really be exacerbated by vaccine hesitancy. And then also we looked at the shape of waning vaccinal immunity. So this is how quickly do you wane, and how does that look over? So you get vaccinated. Now, how does that change over time? Do you wane quickly early, or do you protect? You have a lot of protection initially for a little while before it drops off, and that has a lot of consequences for pathogen invasion. So, a question that has really been I've been really thinking about lately: what about long COVID, and what about other long-term effects? And in particular, what about mortality after recovery? So, a lot of classic models are looking at mortality during infection. How does that impact dynamics? But I think as we see, as COVID has revealed, and I think there's many more pathogens that exhibit this as well, how are long-term effects? What do they have? Like, how does mortality after recovery impact dynamics? And so you can take a simple model and look at this, and basically now we're looking at like post-acute phase, right? And this post-infection mortality, which I call PIM, well, how can you look at that? So you can just split the population into 3 groups, right? They're susceptible, they're infected, they recover. And then you can say they can potentially get reinfected, right? And so it's very simple. It's very similar to what I showed you before. You can have some mortality during active infection, and you can also have this post-infection mortality, and right here, right? This is mortality after you recover. And so to show you the flow diagram, so in our field we like these to illustrate what's going on. It's very similar to what you had before. So there's recruitment, primary susceptibles, they get infected, and they recover, and they potentially get reinfected with potential relative susceptibility epsilon, right? The same kind of parameter as before, but here it's susceptible, infected, susceptible again. So if epsilon is 1, you have no period of immunity, and if epsilon is 0, you have lifelong immunity, right? And then you have this alpha S here. This is this mortality after recovery, right? And so here's the reference. And so this potentially counterintuitive effect is that post-infection mortality can actually trigger sustained epidemiological oscillations. So here's an example. So I discard the initial bit. Now you're looking at the cycle once it's stabilized. And you can see in red IP over time, it's 200 years, right? So it's a long decades of cycles. The fraction of the initial population on the y-axis, the different colors, red I, purple SS, and gray SP, primary susceptibles, purple secondary susceptibles. So what's going on here? So for the mathematicians that are interested, you can plot the eigenvalues of this one pair of eigenvalues that eventually have imaginary parts, and the real part becomes positive. You get this thing called the Hopf bifurcation, which leads to your sustained oscillation. If you push mortality after infection to be really, really high, so effectively individuals are dying immediately after recovery, then that becomes stable again. And if you think about this, you can start to understand where the cycles emerge. So if you consider the R effective, so this is the effective reproduction number. It's basically the basic production number times susceptibles. You have 2 types, primary susceptibles and secondary susceptibles. What infection is doing is moving individuals from the primary pool to the secondary pool, right? And then the mortality after infection, reducing this susceptibles. So all of a sudden, you can reduce your R effective below 1, right? So then the infection goes down. Well, then that allows the primary pool to build up, right? Because we have this recruitment into this primary susceptibility. Then that can, now susceptibles go up, and now you can get R effective above 1 again, and then you get a new epidemic, right? And then you get the same effect. Now you move these back to secondary susceptibles, you die at a slightly higher rate, crash the R effective, and so on and so forth. And then you get these long cycles, right? So because these cycles are so long, if you think about like a short time series of 10 years, it'll be difficult to make, it could be difficult to make inference if you have these kinds of underlying cycles happening. Right? And just to convince you it really is happening, for low amounts of post-infection mortality. So here, just basically plotting the long-term outcome for each value of this alpha S parameter, this post-infection level, as you vary it up over time, right, for different SP, SS, and I. And as you increase this value, eventually you have a stable level, and then boom, your cycle appears, right, at a very low rate. So you don't need much, right? A little bit's enough to trigger these sustained oscillations. And then I did mention that you can have disease-induced mortality, and if you have that, that actually accentuates the effect. So you all of a sudden need less post-infection mortality to get the cycles. And then if you have robust immunity, then that's stabilizing, right? And again, that's intuitive. If you're reducing the pool of the importance of these secondary susceptibles, then you should get less cycles. And so recently, I've also been interested to look at, so this is a very simple model. There's no fully immune period, right? You go from susceptible, infected to potentially susceptible again. What happens if you have a different distribution of the immune period, right? So what if this immune period exists? So you're infected, and then you're recovered, and then you're immune for a certain period of time. And then you become potentially susceptible again. And you might even have mortality during this period of complete immunity, right? So this is the kind of outline, right? So you're susceptible, you're infected, you recover, you might experience this post-infection mortality, and then you wane into the secondary susceptible class, and then you might also experience post-infection mortality there, and then you can get reinfected. The key question is, how does this happen? So to model different distributions, you can just have multiple R classes. So one R class, you're assuming that this distribution of waning of immunity is exponential. If you have more than one, now it becomes Erlang, and you can add them up. And so here's the flow diagram. You can add, so you can have N. You can have as many as you want, right? And then anyways, this is a paper in press. But basically, if N is 1 or 2, so there's R1 or R2, you can show that mortality during infection or after recovery is not destabilizing. If you have post-infection mortality after return to susceptibility, that can actually trigger 3 switches in stability. So as you can really change the dynamics quite a bit, depending upon the duration and shape of immunity. And then the characteristics of immunity and the disease-induced mortality can synergistically complicate epidemiological dynamics. So the answer is it's complicated. So this, I think, hopefully has convinced you that post-infection mortality is a key parameter here, and it's something that's so far been overlooked. And now you can ask, well, how can this mortality after infection ever evolve from a pathogen perspective, right? So pathogen, often there's this trade-off between transmission and the duration of infection. How is there any interplay with evolving to have harm the host after infection, right? After they recover. And so you can model invasion by a mutant that causes post-infection mortality in a simple way. So if anyone's interested, please let me know. We can discuss this further. But essentially, you assume you have an endemic type. So the type that doesn't cause any post-infection mortalities present in the population. Then you say, when can a pathogen that does cause it invade, right? So this is the second pathogen here. And then you have to keep track of all the flows, and things get a little complicated. But you can start to make some analysis and ask, when can you see this evolve? Under what situations? And so what we find is, so this is the sketch again, unlike classic virulence, post-infection mortality is neutral and not selected against. So basically, you can see that already it's more favored, right? That there's almost nothing that prevents it from appearing if you don't have any other trade-offs going on. And if you have a susceptibility mortality trade-off, that can select against classic virulence but favor post-infection mortality. So basically, the answer is this might be fairly easily evolvable. So to study these questions, both from immuno-epidemiological post-infection mortality to these other kinds of post-infection effects, you really need large cohort studies, right? And these can take multiple shapes. Here's one that we proposed. So you have these individuals, you keep track of them as they go through the infection cycle. They're susceptible, they're infected, they recover, their immunity wanes, they can get reinfected. The idea is we want to keep track of individuals as they go through the cycle and keep track of their vaccination type, the sequence of their infections, the serology, et cetera, right? If you keep track of all of this, all of a sudden you know that the person who was infected 6 months ago is getting infected again, and maybe they got vaccinated 2 years ago. Now you have very detailed data on how these immunological responses happen, and in turn, how new pathogens might be affecting that, right? How evolution of new variants may be changing these features So then if you keep track of the household transmission data, then you can estimate the transmission impact of the immune status on the viral variant, right? And obviously, so far, I didn't really mention any age structure, but obviously age structure is very important, right? So we have to monitor across age structures and degrees of immune health, right? So far, there's a key piece, I think, that I introduced and then I never came back to, which is the social dynamics, right? So what about social dynamics, human behavior? That can also influence epidemic dynamics, right? So one of the questions you can address is when is there tension between individuals' costs and societal benefits, right? So we're choosing to adhere to some interventions, we choose to mask, we choose not to mask. Sometimes our individual decision making is what's best for society. Sometimes it might not be, right? It might be what's best for us according to our calculations, but not what's best for society, right? So you can do a simple framework. So this is evolutionary game theory. So I'll introduce it very briefly. Maybe you're not familiar with it, but you'll soon discover that it's rather easy. So you have a matrix, that's a payoff matrix, and N is you're not adhering, and I is you adhere. And so if I don't adhere, and the person I'm interacting with doesn't adhere either, then the payoff is this negative alpha NN capital lambda. So capital lambda is your risk of infection, and alpha NN is how modulated it is by the two decision making, right? And so if I don't adhere, but someone else does, the partner does, then that'll be alpha NI, right? And then if it's the opposite, I adhere and the partner doesn't, then it's alpha IN. And if we both adhere, it's alpha II. And now if I'm adhering, I have to pay a personal cost, right? And that's this gamma I. Okay. So you can see how then you can use this matrix to write a replicator equation which models social learning. And then you can figure out what the long-term outcomes are. And I'll show you that in a second. But what we were really interested in this work is to look at what happens if you have the choice between two interventions. So you can choose to wear a slightly better mask, like KN95 or N95, or you can choose to wear surgical mask, which might have different costs, but maybe also different benefits, right? And so again, here now you have you're not adhering to any intervention, M, you adhere to one kind of intervention, F, you adhere to the other. And then the gamma is you pay different costs for adherence. And then the alphas are, again, you have to think of all the pairwise bits, right? So this is this paper led by Arne Charlson. And what we find is we can actually then do the long-term calculations, and you can look at what happens in one intervention. So I told you can use this equation to model social learning, and you can see what happens in the long term. So on the x-axis, we have this intervention cost divided by the risk. And on the y-axis, it's the fraction of individuals that are adhering to the intervention. So in a high-risk setting, right here, everyone adheres. That's the black line. That's what the outcome is. And that's also what everyone should do, right? So that's a very good. And in the low-risk setting, no one adheres, but also the social optimum is that no one should, right? Somewhere in the middle, it would be better off if everyone adhered, but fewer than everyone do, right? And that's a snowdrift game in this field. So you basically have fewer than everyone adhering, and then a prisoner's dilemma, where no one adheres, but it would be better if everyone did, right? So you can already see that like this is probably where like the nudges and the mandates are most beneficial, right? Where you can really gain at the population level. And then you can look at what happens in the two intervention case. So here we use data from Bagheri et al. to parameterize two different kinds of masks. And now instead of one intervention cost by risk, now we have two interventions, right? Two different kinds of masks. So you have the x-axis is the cost of one kind of mask over the risk. The y-axis is the cost of the other kind of mask over the risk. The left panel illustrates the outcomes. The right panel illustrates the social dilemmas. And you can see the strength of the social dilemmas in the grayscale. So the stronger the social dilemma, the more it'd be better if everyone adhered. And what's interesting here is you can look at all the different kinds of outcomes. So here you have N mask only. Here you have F mask only. Here you have the coexistence of unmasked and F mask, coexistence of unmasked and M masks, no U only. And then here you have a bistable region. So it depends where you start. So depending on where you start, you might end up at one or the other. Now, what's interesting is you never see coexistence of F and M, right? So you never see the two kinds of masks coexisting in the population. It was a little surprising to us, but nevertheless rather interesting. But we're assuming here that we have constant infection levels, right? So this is probably not what's going on, right? We have infections, or you have more adherence, and maybe that influences infection levels, and then maybe that feeds back. So then we decided to try to couple that together and to look at non-pharmaceutical interventions in that setting. So what if you can couple them into this behavioral epidemiological model? So here's what the way we're thinking about this model. So you have these focal players that are adhering or not, and now you make that dependent upon the force of infection. And then the individuals that adhere have to pay a cost. All of a sudden, then you get population of adherers. You know what the fraction of the population that adhere is. Then you compute the average effect of this non-pharmaceutical intervention on transmission, and then you multiply that by the transmission rate in your SIR model, susceptible, infected, recovered, and that gives you an infection level, which then again feeds back. Right? So it's a coupled system, and this is the reference. And then you can look at long-term outcomes. And so here's I'll just illustrate them with an example, and then I'll tell you a little bit more. But if you look on the top, you have the transmission rate and the fraction in behavior, right? So purple is people adhering, yellow is people not adhering. In the bottom, you have again as a function of the transmission rate, the fraction susceptible or recovered on the left, and the fraction infectious on the right. So blue is infectious. Red is infectious, and then blue and gray follow or recovered and susceptibles, respectively, on the left now. So as in the transmission rate, and because here are the parameter values, the transmission rate's almost equivalent to the R naught. So as the transmission rate's low, you have no adherence, you have no disease, and eventually the disease picks up. And you still have no adherence, you have no adherence, and then eventually you have adherence, partial adherence, and then eventually transmission's high enough, but then that triggers total adherence, right? And everyone's adherent. And if you look at this partial adherence region, you see that it's flat, right? So at partial adherence, it looks like the infection levels are independent of the transmission rate. And you can actually show this. You can show this analytically that the infection levels with partial adherence don't depend on the transmission. In the other two cases is just classic endemic equilibria of the SIR model. And then you can reduce endemic levels with vaccination even when there is partial adherence. And here's the example. So you can see this partial adherence, you reduce the level for different values of P. The level of infections does go down even at partial adherence. So what's going on here? So the idea is you have compensation, and behavioral compensation can basically fully compensate the changes in the R naught, in the transmission. They increase transmission, more people adhere, and therefore you have the same level of infections. You reduce the transmission rate, fewer people adhere, and you have the same level of infection. So it's good to keep in mind that when you have different interventions, these things can interact, right? And so when you're thinking about intervention design, this is potentially relevant. And then, don't have time to discuss this, but Carl Bergstrom and Bill Hanage wrote this great commentary that really highlights this work and this contextualizes it. And so if you're interested to read this, I would encourage you to look at this work as well. And so now in the last minute or so, I'd like to move into this idea of like, now we use behavior, we know behavior can influence infectious disease and vice versa. Can you now use animal behavior to try to help anticipate or predict the zoonosis? So with Wayne Getz, we wrote this commentary, and we're pretty excited about it, about complexities of One Health. And the idea here is that we have this conceptual One Health early warning signals approach, and we are thinking, or we're proposing that potentially you can use passive monitoring of animal behavior, movement, and temperature with some kind of AI system to detect changes and then know that maybe there's a pathogen coming. And obviously this rather difficult, but maybe if we identify certain indicator species to monitor, this can help us in this direction. And obviously, as the world is changing, this becomes really relevant. And so to conclude this talk, I'd like to go back to the initial flow diagram, or a bit different, but the same kind of idea, right? So we started off talking about emergence and transition to endemicity, and hopefully I've convinced you that the strength of immunity is a key parameter here. So there's some potentially important counter or unintuitive interplay between immunity, non-pharmaceutical interventions, and seasonality. And I've brought you forward to post-infection mortality triggering cycling. I hope I've convinced you that this is important, and that post-infection mortality can readily evolve, and that those vaccine dosing regime can influence evolutionary potential. And then I moved into large cohort studies and highlighted that they're needed to study immuno-epidemiology and evolution. And then we looked at individual decision-making, social dilemmas, highlighted that social dilemmas can emerge, and then that behavior can fully compensate changes in transmission. And then I've presented to you this idea of monitoring animal behavior for emergence of new disease. So we're back. So thanks to the co-authors, and thanks to you all for listening, and I'm ready to take some questions. and meet. We can stop the Zoom now. We can stop the recording. Oh yeah, awesome.